Hello, my name is Justin Lerma with the University of Texas, Rio Grande Valley, an undergraduate senior with the biology department. Today, I'm going to talk about the small project looking at the below-ground microbial communities correlating with above-ground plant growth. Recently, the soil microbiome has received research interest in agricultural fields as a method to increase crop yield. The project set out to study the microbial hotspots that develop from soil abiotic conditions and plant feedback, and the factors that can determine them. We conducted the study in an organic grain farm in South Texas, which is a semi-arid subtropical climate. Soil samples for this project were collected from a 15-acre field, and the sites of interest were identified using an aerial drone. The field was mapped with microsites based on canopy cover. Sites with less than 25% canopy cover were recorded as low, 25 to 50% as medium, and more than 50% as high. We collected soil samples at a 10-centimeter depth and analyzed for chemistry and biology. We looked at organic matter, moisture, pH, soil temperature, and soil respiration on site. For the biological parameters, nematodes were extracted from the soil and identified, and soil samples were sent for PLFA sent to the USDA ARS lab of San Joaquin Valley in California. As well, the plant trait characteristics of Pseudomonas fluorescens from the sun hemp used in the experiment as the main cover crop were done at UTRGV in an interest for rhizosphere engineering to help solve the soil-specific problems of the Rio Grande Valley, as the soil of South Texas lacks ammonia, phosphate, and zinc, of which the hope for Pseudomonas fluorescens was to help promote these important plant traits. Moving on to the chemical methods, the soil moisture was collected with the gravimetric method, and the organic matter was determined with the combustion method. pH and salinity were recorded with a pH meter from a mix of soil and DI water. Total carbon and nitrogen were determined with the combustion method on a leukoCN analyzer, and PLFA results from the field were freeze-dried and sent to Dr. Lauren Hale of USDA ARS. For nematode extraction from the soil samples, we use the Jenkins method, which is a rapid centrifugal flotation technique for the separation of nematodes. Soil samples are sieved to remove organic matter and are subjected to rounds of centrifugation, and sucrose is added to separate the nematodes from the soil. Nematodes were identified by feeding groups based on mouthparts, as shown in the picture on the right. Based on the data collected and analyzed, the relative abundance of the soil nematode community and the microbial biomass based on the PLFA results showed no significant difference among the different sites in the agricultural field. After the non-significant results of the microbial community, interest in the lab grew in the rhizosphere of the sun hemp used in the experiment. The soil of the Rio Grande Valley lacks ammonia, phosphate, and zinc, important plant growth promoting traits. Normal ammonia values range from 0.1 to 1%. The amount of ammonia in the soil of South Texas is less than 0.001%, far less than the average. As well, the pH of the soil is normally high, making phosphate and zinc unavailable to the plants. Pseudomonas fluorescens was targeted as the main plant growth promoting rhizobacteria of interest, as it is easy to culture with a relatively fast growth rate. Pseudomonas fluorescens creates enzymes that help convert the inorganic forms of zinc, phosphate, and ammonia into the organic forms beneficial to the local soil, and thus may help promote crop yield if used as an alternative to fertilizer, which creates runoff of excessive chemicals, which damages local native flora and fauna. However, after the identification of Pseudomonas fluorescens in the sun hemp field, the three different growth categories show no difference in the percentage of plant growth promoting traits that Pseudomonas fluorescens tested positive for in the laboratory setting, meaning that there was no statistical difference for the percentage of Pseudomonas fluorescens strains that tested positive for the plant growth promoting traits among the three growth categories. The soil chemistry results showed no significant correlation on the different variables that were analyzed except for soil nitrogen and soil carbon. Here I'm showing the total carbon on the bottom and the total nitrogen on the top for each growth category. Both values were significantly higher in sites with high plant growth. As well, interestingly, soil respiration was highest in medium growth, with no significant difference between high and low. In conclusion, there was no significant difference in any soil biological parameters except for soil respiration, which was highest in medium growth. As well, total soil nitrogen and carbon were high under high plant growth, with no statistical difference was found in any other soil chemical parameters analyzed. And so high plant growth was not an indicator of microbial hotspots in this project.